Chapter 19, Theory Video 4, Vectors. Now, vectors, when we spoke about diseases, we spoke about vectors who carry the disease from one person to another. So, just like that, in this particular scenario, we are talking about gene vectors, that is who carries the gene from after genetically engineering the gene, how do we transfer it to a cell? Who carries the gene into the cell is it would be defined as a vector. So there are three different kinds of vectors that we spoke about: viruses, plasmids, and liposomes. So now we're talking about plasmids. So plasmid is a circular piece of double-stranded DNA. What we do is, when it comes to plasmids actually occur naturally in bacteria, and most of the time they contain genes against antibiotics, or the genes for antibiotic resistance. So what we do is, we use one of the qualities of these plasmids, that is the ability for these plasmids to be exchanged between individual bacteria, and it could be even between different species. So what we do is, we cut this plasmid using the enzymes that we spoke about. The restriction enzyme that we use to, the same enzyme that we use to cut the gene that we are going to use, we cut the plasmid, and then so that when we cut it with the same enzyme, it will be the sticky ends are going to be complementary. So however, if there is a blunt end, if the restriction enzyme will give a blunt end, then we will have to add two sticky ends, so that it would be that is to the gene, and as well as the plasmid, we will have to add two sticky ends so that it would be complementary. So so the process happens like this. So there is a bacterium. Inside the bacterium, you have the plasmid. How do we get this plasmid separated? What we do is, we use a centrifuge to separate. After breaking down the cell wall, we use some enzymes to break down the cell wall, and after that we rotate it in a centrifuge. Then what happens is, the larger circular DNA will be separated from the plasmids, because plasmids are small. Then we can get these plasmids into our use. So the bacterium has plasmids, we treat it. Then in this particular situation, we have cut it using BMH1. So when it reaches this point, it has been cut. So at this point, it has been cut, and then what we do is, we use in the same BMH1 cutting mechanism. We cut the gene that we require as well. The human gene is cut, and then what we do is, we insert it into the when we mix it, it will be the plasmid will accept the complementary base pairing base, and it will pair up. Then we treat it with ligase enzyme, and the recombinant plasmid, bacterial plasmid with a human gene would be given out, right? So this is what happens. So the function of the ligase is to it will link together the sugar phosphate backbone that was cut in the early operation. It will rejoin it with the plasmid, and as a result, we will get the final product. How these ligases do this, that is the joining of the backbone, is by catalyzing the formation of phosphodiester bond. So that provides a closed circle of double-stranded DNA that contains a new gene. As a result, the recombinant plasmid is formed. Now, we can not only obtain bacterial plasmids, but also we can produce them artificially. So there is a group called PUC group of plasmids. They have a low molecular mass, so they can be taken up by bacteria quite readily. Then the origin of replication, so they can be they have an origin of replication, so they have the ability of being copied. Then they have several the unique target sites for different restriction enzymes in a short length of DNA, and it is called polylinker. Then there would be one or more marker genes, so it would be easy for us to identify after it has been taken up by the plasmid. So after producing the plasmid gene link, now our purpose is to transfer this plasmid into the bacteria. First of all, what we need to do is the bacteria and the plasmids need to be put into a solution with high concentration of calcium ions. After we mix it, we have to cool it, and later on, and we should give it a heat shock. So this allows, or this actually increases the chances of plasmids passing through the surface membrane of the bacteria. So it is cooled and then given a heat shock. So that contraction and expansion allows the plasmids to pass through. But we have to remember, not all the plasmids, not all the bacteria would take up the plasmids. Only about 1% will take up the gene containing plasmid. So the rest will take up plasmids that would have not joined with any gene, or would not take any plasmids at all. Right. So now we need to identify the bacteria with the recombinant DNA, because we need to copy that. So whether we have to figure out whether the bacteria has taken up this taken up the gene, we need to figure out we need to figure this out, because that is the gene that we require. Earlier what they did was they spread the bacteria on an agar plate which contained an antibiotic, because when it responds to the antibiotic, they would know which bacteria would contain it. However, now there are better methods that have been developed. So obviously we use genetic markers to do this. DNA polymerase in bacteria will copy the plasmids, and then bacteria will divide by binary fission. So each daughter cell will have several copies of the plasmid. So when you produce many copies of the recombinant plasmid by gene cloning, we produce multiple copies of the gene that we produced as a result of genetic engineering. So the bacteria would also transcribe the new gene and use mRNA in translation to form a protein known as a recombinant protein. So that is as a result of the formation of the gene. I hope you understood. If you have understood, please do the questions 3, 4, 5. If you do not understand, if you need further clarification, please let me know. Write down a note on this. Vectors carry the gene to the plasmid. I'm sorry, the vectors carry the gene to the cell. We discuss about three types of vectors: plasmids, viruses, and liposomes. When plasmids are used, they are cut with, their circular strands are cut with the same type of restriction enzyme used to cut out the gene from the human chromosomes, because this would produce sticky ends which are complementary. If a blunt end is produced, sticky ends would have to be added to the plasmid as well as the gene. Then it would be mixed and treated with ligase enzyme, which would reform the backbone phosphate backbones of DNA by catalyzing the formation of phosphodiester bonds. Leave a line and write: plasmids can be modified to produce good vectors and also can be made artificially. PUC group of plasmids have, just write it as a name and remember it as a name. Number 1, a low molecular mass would be taken up readily by bacteria. Number 2, an origin of replication, so they can be copied. Number 3, several single target sites for different restriction enzymes in a short length of DNA called polylinker. Number 4, one or more marker genes allowing identification of the cells that have taken up plasmids. Leave a line and write. After plasmid has taken up the gene, it would be mixed with a solution of high concentration of calcium ions Mixture is cooled and then given a heat shock. This results in about 1% of the bacteria taking up plasmids with the gene and are transformed. To identify transformed bacteria, we use genetic markers. Leave a line and write DNA polymerase in bacteria will copy the plasmids. When the bacteria divide by binary fission, many copies of recombinant plasmid would be produced by gene cloning. And I hope you understood. Please do question number 3, 4 and 5.